All right, this week we're talking Alzheimer's disease, and I thought I'd start off with a little bit of background about how Alzheimer's forms from a biochemical level and how it's thought to be, in many ways, kind of like a prion disease. So as a background to this, the concept of a prion came from diseases like Kuru, mad cow disease. There are many, many others that spread around animal populations and some very rare ones in humans. And the idea is that every protein, in order to be functional, has to be folded in a particular three-dimensional conformation. And so there are proteins, confusingly, there's one actually named prion protein that's involved in mad cow disease. In humans, it's called Creutzfeldt-Jakob disease. And there are many, many others, like amyloids and Alzheimer's and alpha-synuclein and Parkinson's, where the protein can take on different shapes. And so in this model, what we're looking at is this cube and this red pyramid are supposed to represent the exact same protein, just different shapes of that protein. And so what's unique about prions is their ability to cause other proteins to change their shape. And so in this refolding model, I like to call this the exposure model, because the idea is everyone has these normal, healthy proteins in their brain that have the correct shape, right, illustrated by the blue cube. And when you're exposed, right, there's some sort of exposure. In Kuru, it came from the ritual eating of brains of people. And if their brain was infected with the Kuru prion protein, then getting that protein into your system would allow direct contact between the prion form and the healthy form, converting the healthy form of the protein into a prion itself. And this basically leads to a snowball effect where the more prion proteins you have that have the prion shape or conformation, the more they can bind to and convert the healthy proteins into even more of the prion form. And pretty soon they spiral out of control and you get these big masses of protein that are called plaques. Okay. Now, in Alzheimer's disease, it's very, very unlikely that there's any sort of exposure going on, because how would you be exposed to the brain of an Alzheimer's patient? It's just not realistic, outside of some sort of crazy examples like dura mater transplants, transplanting brain tissue from a deceased donor who had early stage Alzheimer's. And in that case, it can happen, transmitting Alzheimer's prion proteins into the brains of healthy people and spreading the disease. But most people with Alzheimer's get it spontaneously. So that's this idea of a seeding model or an equilibrium model where something allows the healthy form of the protein to convert into the prion form, and then possibly it can convert back. I don't really buy the converting back part, because these proteins are incredibly difficult to get to convert back from the prion model back to the healthy form. But the concept is, in healthy people, most of the time, this conversion doesn't happen. You have your normal healthy version of this protein. It doesn't take on the prion form. And then for some reason that's not really all that well understood, some of them start converting into the prion form. And once you get those first seeds, or this is the seeding model, then it can continue to spread by converting even more proteins to the prion form, right? Because the healthy brain is full of the original protein that's folded correctly. And now that we have these so-called seeds, they can convert even more and more. And we wind up with the same thing, these plaques or amyloid plaques that form, that are really one of the key defining hallmarks of Alzheimer's disease. All right, and so you have many different prion diseases. In Alzheimer's, you wind up with two major protein aggregates that result from some kind of prion activity. There is amyloid beta plaques, right, which are extracellular. They're outside of neurons. They're enormous, many times larger than an individual cell. And they've gotten most of the attention when it comes to Alzheimer's treatment because, quite frankly, they were the easiest thing to observe under a microscope 100 years ago. And so they sort of, became the defining characteristic. But equally, if not more important, are structures called neurofibrillary tangles that are made from a protein called tau. They're intracellular. So these black stain structures, this is histology from an Alzheimer's brain after an autopsy. Notice the plaques are just big masses. They look like big balls of protein. But the tangles, if you zoom in close enough and maybe connect a few dots that are missing because of the plane of the cut, they look like neurons. And the reason for that is tau is intracellular. It builds up inside of neurons. And then eventually the tangle is really just the hollowed out skeleton of that dead neuron that was filled with an overabundance of tau, right? And so the end result in this is your neurons are dead, and how much of it is because of the plaque and how much of it is because of the tangle has been a bit of a debate. But I am very firmly in camp tangle, that the tau is really the cause of neuronal death. And do the amyloid proteins have something to do with triggering tau and making it accumulate? Yeah, probably. But a lot of the drugs that have targeted amyloids haven't really worked all that effectively. And part of the reason is once you have plaques and tangles, the damage has already been done. These neurons are toast. They're not functioning anymore. And there's no way to get them back because in an adult brain, very, very few neurons are capable of dividing, and you're just not going to get that population of cells back again. Okay, so a little bit more on amyloid because that's what the drugs that we're going to be talking about are targeting. So amyloid beta, A-beta, is actually a very, very tiny fragment. So this is the A-beta peptide that's released from a larger protein that got its name, APP, amyloid precursor protein, because it's exactly what it is. Every healthy cell in a person's brain makes that entire APP protein. It's inserted into the cell membrane of neurons, and there are a handful of enzymes called secretases, right? Alpha, gamma, and beta secretase. They cut the APP protein. If you get a cut by alpha and gamma secretase, you get this harmless little fragment called P3 that floats away and does a whole lot of nothing. If you get a beta gamma secretase cut, you get the A-beta fragment. That is the little chunk of protein that aggregates to become plaques, right? Amyloid plaques inside of an Alzheimer's brain. And so we know that this is definitely tied to Alzheimer's in key ways because certain mutations in APP or in the beta secretase gene that make it hyperactive lead to more A-beta production and higher risk of Alzheimer's. Those are some of the ways that people can get familial, like genetically inherited Alzheimer's. And so it's clear that amyloids play a role, but the question is at exactly which stage of the aggregation pathway are they important? So every amyloid protein starts off as a monomer, meaning an individual little protein, tiny, submicroscopic, beyond what you can even see with an electron microscope. And eventually, those little individual monomer units start pairing into dimers, sets of two, trimers, tetramers, pentamers, and they get larger and larger because they're sticky, and that's part of their prion-like function. Eventually, those oligomers will start binding together and form seeds of what become fibrils, and fibrils are basically strands of these amyloid proteins that wrap around each other and are braided almost like ropes. Okay, so you have this progressive growth from monomer to oligomer to protofilaments to fibrils, and eventually fibrils form these big, massive clusters called plaques, where there are trillions of proteins, of individual peptides in a single plaque. They're huge structures on a cellular level anyway. Okay, so the question in all of this is, where is the damage coming from? Is it monomers that are toxic? That's unlikely because even healthy people have amyloid monomers floating around in their brain. Is it the plaques? Is it the fibrils? Is it the oligomers? There's a lot of data on this that I don't really have time to go into, but there is very, very strong evidence to suggest that the oligomers are actually the most toxic form of amyloid. And to date, most of the drugs that have targeted amyloids are removing fibrils and plaques, and some of them may have some reactivity with oligomers as well, but it's not as strong. And my take on this is that we would be a lot better off targeting the oligomers because by the time you get to this fibroplaque stage, it's probably too late. But we'll probably find that out as more antibody-based amyloid medications are developed that target these different stages. It just gets difficult because there are many different sizes of oligomers and no one antibody is likely to bind effectively to all of them. All right. Another line of evidence for supporting the fact that both amyloid and tau are somehow involved with each other and that there has to be an interplay. It's not one or the other. They interact in some way. This study took CSF samples, right, cerebrospinal fluid, from a bunch of different people who did not have Alzheimer's yet, but were at high risk because of family history. And they measured a lot of different variables, and they followed these people over time for more than a decade to see who developed Alzheimer's, who developed MCI as mild cognitive impairment. That's sort of a precursor to what will likely become Alzheimer's eventually, but it's not diagnosable yet, and who remain normal. And what they found was if you measure amyloid levels, right, given as A beta is amyloid beta, 1 to 42 is a specific variant of the amyloid peptide found in CSF, and you measure a particular version of tau that's been phosphorylated at a specific amino acid, and you graph them, what you find is it's not perfectly explanatory, but people that are normal are more likely to fall in this region, relatively high levels of amyloid, low tau. People that have Alzheimer's are more likely to fall in this region, low amyloid, higher tau. And the same thing goes for MCI. If you follow those people further, it's very likely that some in the normal group are eventually going to get an MCI diagnosis, and some people with MCI are going to be progressed to an Alzheimer's diagnosis. So the point is, we can somewhat predict who's going to get Alzheimer's by taking these CSF samples, but it's not perfect. We can't predict the timeline, but maybe this gives us an idea of what patients could participate in clinical trials where Alzheimer's prevention is the goal, which to this date hasn't been done yet. So far, we've only tried Alzheimer's drugs on patients that already have a diagnosis, but they don't work that well. And so the hope is maybe we can take people that are currently normal, but if they fall in this range of amyloid and tau levels, that maybe down the road, we can prevent them from getting the Alzheimer's diagnosis that they would have got without a treatment. Okay, so how are we going to tackle this? If you want to remove amyloid plaques from a person's brain or any species of amyloid, the only realistic way to do it is to go through the immune system, because there's no drug that is going to bind to these amyloids and inhibit them. They're not receptors, they're not enzymes, they're not really a normal drug target. But if we make an antibody and convince that antibody to bind the species of amyloid that we're interested in, then you can get the immune system of the patient to go after that amyloid and remove it through phagocytosis. So this is a method that's been used a lot for cancer, and the idea is in a cancer treatment, you find an antigen on the cancer cell that is unique to the tumor, right? In the amyloid version of this, the same idea applies, but instead of an antigen on a cancer cell, we want to target a very specific version of the amyloid peptide, a fibril, a certain size oligomer, a plaque, whatever it might be. Once we have that antibody in mind, the only way to make it is to use an animal. And so we're going to use a mouse or a rabbit to produce antibodies against those proteins, because they're human proteins. They'll be seen as foreign by that animal, right? And then we need to effectively humanize the antibody to turn it back into having at least some of the original human sequence that the human immune system will recognize, so that the patient's white blood cells don't see it as a mouse or a rabbit antibody, right? And once we have that, right, we inoculate our mouse with the kind of amyloids that we want the antibody to stick to. We then take cells out of that mouse's spleen that can produce those antibodies, turn them into a hybridoma, which is a type of cell that basically becomes an antibody factory. And we screen them for which ones are going to be best at binding the amyloids that we're interested in. Right? And then that antibody is used as the actual medication. Right? So whenever you see these drugs that are in trials, or some have been approved for treatment for Alzheimer's, that suffix, that ending MAB, stands for monoclonal antibody. That means that they target a specific type of amyloid, right? And so some of these drugs are targeting amyloid monomers and small oligomers. Some of them are targeting fibrils. Some are targeting protofibrils. Some of them are targeting actual plaques. So far, the drugs that have actually been approved for at least temporary early stage use in Alzheimer's, Donanemab, Aducanumab, and lecanemab are the three that have received some sort of approval. And there are more in trials. I'm guessing we're going to get more approvals in the future, and we need to tease out which ones are most effective and if maybe some of them are effective in combination, right? My inclination is that we're probably going to see the greatest effects from drugs that target these earlier stages of formation, but that remains to be seen as far as human trials go. So let's run through a few examples. Aducanumab is one of the antibodies produced by Biogen, and if you circle back and notice, its major binding is to plaques, but these lighter gray arrows are showing that it does have some affinity for fibrils and oligomers as well. So take that into a clinical trial where, unfortunately, this was a clinical trial where the patients that were tested were relatively late stage, but we're trying to see if we can slow down the progression of their disease. Right? And step one is finding out, okay, who are we actually working with? So the sample size on this was substantial, but not incredible, right? Approximately 45 to 50 people in each group that either got placebo or the actual drug, and they tried multiple doses, right? This is the dose of antibody in milligrams per kilogram, and then another dose where they slowly ramped up. So titration means the dose started low, and then they eventually increased to have the maximum dose. One concern that's followed a lot of these drugs is they can target vasculature because amyloids stick to blood vessels in some cases. And so this brain microhemorrhage is one of our biggest concerns. Notice this happened in 3 out of 46 placebos and started happening in progressively larger numbers, unfortunately, as the drug was increased. And so this is one concern that has sort of dogged these therapies a little bit is we know that this is a risk because blood vessels are permeated with amyloids, and it's hard to avoid that. I think the hope is if we can use the drugs against or for patients in earlier stages of the disease that don't have as much vascular damage, the hope is that the benefits will increase and hopefully the side effect will decrease. Right, but the good news, there were a few notable effects. The first was there was a significant reduction in amyloid burden. Right, so this measurement, it's not critical exactly how it works, but this is effectively a measure of amyloid burden, plaque levels within these people's brains. And this gray line was a placebo for the first 12 weeks of the study. And then after that, since significant reductions were seen across the board, the placebo was discontinued and all patients received the antibody. And you notice there was no decrease in the placebo during that introductory period. And then throughout the entire study, there were significant reductions at every dose. And importantly, because it makes you feel a little bit better about the mechanism, the reduction was the greatest for the higher doses of the antibody. So we got a pretty significant reduction in plaque load. And the next parameter that obviously people care about the most is what is the change in cognitive symptoms? So this is a measure of a clinical dementia score, which is a multi-part screening to assess the patient's cognitive function, memory, a lot of things that go into an Alzheimer's severity diagnosis. And interestingly, we got reductions almost across the board, meaning all of these patients were worse after 48 months of therapy. And so none of these medications prevented them from declining completely, but they did slow the decline, right? So our best outcomes, right? These two groups showed the slowest rate of decline, which was encouraging because those were the highest doses of the drug, either the titration that was the ramp up or the high dose from the start, 10 milligrams per kilogram. So at this point, we've got a drug that does not reverse Alzheimer's. It doesn't even prevent the continuation of decline in Alzheimer's, but it slows that decline substantially, which, isn't the home run that everyone would have hoped for, but it's the first time it's ever been done. All right, so we finally have a drug that it can at least slow things down a little bit, which gives some hope for future development. All right. Okay, the other Biogen antibody, lecanemab, same idea, clinical trial. What's interesting is this is being trialed in early stage Alzheimer's disease. This trial was just from last year, so it's fairly recent. And what you see is we get a decent reduction in a lot of different measures, right? A dramatic reduction in amyloid burden versus placebo, which tells us that mechanistically the antibody is doing exactly what we want it to be doing. Now, when you compare that to these cognitive scores, panels C, D, and E are different cognitive scores. The placebo versus the drug, there clearly was a difference. The difference was significant in all cases, meaning that lecanemab slowed down cognitive decline, but being completely honest, it wasn't really all that dramatic. So it was statistically significant. The drug is clearly having an effect, but I think one of the major takeaways from this is the difference between placebo and drug for amyloid clearance was enormous. The difference for cognitive measures was pretty modest. And so I think that we have to take this to mean amyloids clearly have some impact on cognitive decline in Alzheimer's, but it definitely isn't the only thing that matters. And clearing plaques probably isn't going to be enough on its own. Okay. And then, of course, there's always the safety question. So this is a data table showing lecanemab versus placebo for a handful of adverse events. And when they screen for anything that is, expected to be potentially a result of the treatment, there were a very significant increased number of adverse events for the drug compared to placebo for a host of different things, but many of them centered around reactions at the site of infusion, which isn't really unexpected and not that big of a deal. This micro hemorrhaging is a much bigger deal. And the number of people that it's affecting is substantial. If you add them all up in total, notice that effectively double the percentage of patients had microhemorrhaging of blood vessels compared to controls. And so this is an issue that's going to have to be worked around. But at this point, it seems that the consensus of the field is, even with the adverse effects on blood vessels, it's worth pursuing just because this is the only real lead that we've ever had for reversing or preventing the advance of Alzheimer's. Okay, last one, Donanemab. This is made by Eli Lilly. This is the only version of this trial that I could get a hold of. The PDF was behind a paywall. But again, early symptomatic Alzheimer's disease, which is, it's encouraging to see trials happening in somewhat younger early stage patients. They had a massive sample size, right? Over 8,000 people. A decent number were excluded for a handful of different reasons, but we still wound up with over 1,700 individuals that were randomized into receiving either the antibody or the placebo, right? And a substantial number of those that actually completed the trial so we could get significant data. And again, the results were significant but not dramatic, right? In every case, they're splitting these people up into levels of tau that they have and a handful of other populations and measuring adjusted cognitive scoring numbers. So basically, how are these people progressing based on how long their diagnosis had been going on? And what you see is, just like before, the patients on the antibody therapy across the board perform better, but it's not dramatically better. We're not really stopping the progression. We're just putting the brakes on a bit and slowing down that progression, which is progress, but it's not enough to really leave anyone feeling all that satisfied. All right. As always, hope this was interesting, and we'll dig into more details soon.